Okay, so today's topic is Streptococci species from veterinary microbiology. So Streptococci are gram-positive. These are cocci, as the name suggests, cocci, spherical, diameter 2 micrometer, non-motile, that is they do not have any flagella. Do not have any flagella. Any bacteria which is motile because of any kind of flagella, but because only of and only because of flagella. Okay, since they are non-motile, so these do not have any flagella. Facultative anaerobes. Facultative anaerobe means their preferred environment for living is in the absence of oxygen, but they can also survive in the presence of oxygen. Okay, catalase negative. Catalase negative, that is they do not have the enzyme cytochrome C. Oh, sorry, catalase. So I forgot the name of the enzyme. Sorry. Okay, next, it causes pyogenic infections. So pyogenic infections means it causes pus forming infections. Now habitat, so commensals on mucosa of upper respiratory tract and lower urogenital tract. Commensals means a type of symbiotic relationship. It lives without harming the host until its number is in control and there is no injury caused to that mucous membrane. Only till then. Otherwise, if its number exceeds the normal limit of the bacteria and microflora, and there is injury to the mucosa, then it will cause harm to the host. Okay. Now differentiation of streptococci. So there is a property of streptococci that is called hemolysis. Hemolysis, hem means blood or RBC, and lysis means breakdown. So hemolysis. So it is of three types: beta, alpha, gamma. So it is just opposite. This is just the opposite of hemolysis pattern obtained from staphylococci. So this is streptococci which we are studying. So it is just opposite of staphylococci. So here gamma, it is same as in case of staphylococci. No observable changes in blood occur around the colonies. Alpha here is partial or incomplete, and beta here it is complete hemolysis. So this is the opposite of staphylococcus. Hemolysis produced by staphylococcus. Okay. Next, pathogenesis and pathogenicity. So if we talk about the pathogenicity, then these are pyogenic organisms, that is pus forming, abscess formation, that is pus formed vesicles, septicemia. Septicemia is a clinical compounded state in which multiple disease causing organisms are present in the blood, and they produce clinical signs. So that is the meaning of septicemia. If we talk about Streptococcus suis, however, so it is pyogenic, it is a major pathogen for pigs, and it causes septicemia, meningitis, and pneumonia. And later we will study that meningitis is the characteristic clinical sign produced by Streptococcus suis. Okay. Now, beta hemolytic streptococci are more pathogenic than alpha hemolytic. What is the meaning of this statement? This statement means that beta hemolytic streptococci are more harmful for the animals than alpha hemolytic. Why? Harmful for humans or animals. Why? This is because of the fact that beta hemolytic are the complete hemolytic means that they produce complete hemolysis, that is completely destroy the RBC. So that is why they are more harmful than alpha hemolytic. Now valence factor are enzymes and exotoxin that is streptolysin. Diagnostic, using PCR, using isolation by isolating the organism. Okay, no issues. Next is clinical infection, important. So first is strangles. So strangle causative agent is Streptococcus equi. It is an infectious as well as contagious disease of horses. Okay, infectious means it is transmitted by microbes, pathogenic microbes, and contagious means it is transmitted by direct contact. Okay. Next, it is febrile. Febrile means that it involves high temperature, fever. It involves the upper respiratory tract with abscession of lymph nodes. So it involves upper respiratory, as we know that these bacteria, the commensals on upper respiratory tract, mucosa, upper respiratory and lower urogenital tract. So these also cause abscession of the lymph nodes. Now pathogenesis and pathogenicity. So this is the horse. So H. equi enters the horse body through the tonsils and then extend to the lesional lymph nodes. So these are tonsils. The H. equi will enter through the tonsil through inhalation mode. Inhalation, the H. equi will enter through the tonsil, enter into the tonsil, and it will affect the regional lymph node. Now what will happen? This bacteria multiply in the regional lymph nodes, and this leads to inflammation, outpouring of neutrophil, and abscess formation in these lymph nodes. So there will be inflammation, so neutrophil will be outpoured, and abscess, that is pus formed vesicles, will be formed in these regional lymph nodes which are present adjacent to tonsils. Okay. Now next, large number of neutrophil reach to the tonsils where S. equi is replicating. This is not for use. So now S. equi has many virulence factor, and these virulence factor protect the bacteria from host immune system. So what are virulence factor? We are hearing this term again and again, virulence factor. So virulence factor means any strategy or product which is produced by the microbe or bacteria. In this case, which causes damage to the host organism. Okay, host. So there are two valence factor in this case in Streptococcus equi: hyaluronic capsule and M protein. So hyaluronic capsule and protein, they have both combined, they have three functions. Protect the sequoia from being phagocytized, they produce a leukocidal toxin, and they prevent the activation of complement system of host immune system. Let us try to understand these 3 points. So capsule specifically, hyaluronic capsule specifically helps in this point, this property: protect the sequoia from being phagocytized. These two produce a leukocidal toxin, that is leukocytes, that is WBC killing toxin, and they prevent the activation of complement system of host immune system. Okay, what is complement system? Complement system is nothing but a part of innate immune system in which has the ability to help the immune system in identifying the various foreign particles or antigens in the body and to kill them. Okay, okay. So this we have studied, not of our use. Now clinical signs. So incubation period is 3 to 6 days. Febrile, high fever, depressed, that is low body function, anorexia, that is low appetite. So febrile, depressed, anorexia, these three are the clinical sign of every disease. We can write this in any disease you want. Febrile, depressed, anorexic. Now oculonasal discharge. Oculo means eye and nasal means nose. So oculonasal discharge, that is discharge from eye and nose, which later become purulent, that is pus, infected with pus and yellow green color. Bacterially infected pus for pus. It also contains pus. Okay. Now characteristic, submandibular nodes are affected. Submandibular nodes, that is lymph node, the submandibular, that is the nodes present at the junction of lower jaw and upper jaw. And then they rupture and they release the purulent infectious material. Okay. Guttural pouch emphysema is also seen, that is accumulation of pus in the guttural pouch. So this is, guttural pouch is present only in equines. So guttural pouch emphysema, that is accumulation of pus, is seen in equines. Okay. Now death occurs due to death in equines occurs due to complication, that is pneumonia, neurologic, and asphyxiation. Asphyxiation, that is deprivation of CO2. Okay. Now mortality rate, morbidity rate, not that much important. Mortality less than 5%, morbidity rate 100%. Diagnosis. Now there are two types of diagnosis: presumptive or tentative, and confirmatory. So presumptive or tentative, when an healthy animal is exposed to infected animal. Simple. An healthy animal is exposed to infected animal. So this is the meaning of the fact that yes, this is a kind of presumptive or tentative diagnosis. Yes, we can say that this animal which was in contact with the infected animal might have become infected. Okay. And confirmatory diagnosis, isolation of S. equi. So we are able to isolate this bacteria from the equine sample. So that means it is a confirmatory diagnosis. So what are the characteristics of colonies produced by S. equi? They are mucoid, diameter is 4 millimeter, and they are surrounded by a wide zone of complete hemolysis, that is beta hemolysis. Okay. Now treatment and control of strangles. So administration of penicillin to those horses which are infected and in contact with infected horses, obviously. So we should administer penicillin. Penicillin should be administered before the development of lymphadenopathy. Lymphadenopathy, that means the inflammation of lymph glands. So before the development of lymphadenopathy, penicillin should be administered. Clinically suspected animals should be isolated. Horses should be isolated for 2 weeks. Types of vaccines: live attenuated and live attenuated intranasal vaccine. Okay? So these are two types of vaccines. Okay, let's move ahead. Now let us move on to Streptococcus suis infection. So suis means swine. So causative agent Streptococcus suis, infection of swine. Now general clinical signs are meningitis, arthritis, septicemia, bronchopneumonia, sporadic cases of endocarditis, neonatal death, and abortion. So sporadic means occasional cases. Now biochemical characteristics, this is very important. There is a complete table given in Quinn and Carter. I will also upload that in my lecture, in some lectures. So that is the Lancefield classification. So Streptococcus suis properly belongs to Lancefield group D, and this is a classification of Streptococcus based on the carbohydrate antigen found in their cell wall. So this point is extremely important. Okay. Not that serological testing is based on antigenic differences in okay, not of importance. Now clinical signs, asymptomatic carrier pigs harbor the SCVs in their tonsillar tissues. Disease outbreak are more common in intensively reared pigs. Now what is the meaning of this statement? Intensively reared pigs. See, the present system of management of the farm animals or the animals which are used for meat is the intensive system of management. That is, intensive means modernized and mechanized system of management. That means that the system is, there is overcrowding, there is no ventilation, there is no sunlight, there is poor hygienic conditions. So all of that creates stress condition on the animal, and that makes them vulnerable for diseases. Okay, so predisposing factor we have discussed. Sows, carriers. Okay. Now meningitis, as we have already discussed in the beginning in the introduction of this bacteria, meningitis is the characteristic clinical sign of Streptococcus suis infection. Meningitis is fatal. There is inflammation of meninges of brain and spinal cord. It is characterized by femur tremor, incoordination, opisthymos, and convulsions. Okay, so this is not of our use. Okay, control penicillin and ampicillin can be given. Okay. Okay. Now we'll move on to the last topic of streptococcus, that is infections caused by streptococcus, bovine streptococcal mastitis. So as we know, mastitis is a multifactorial disease, that is, it is caused by multiple bacteria. It is caused by streptococcus, staphylococcus aureus, it is caused by streptococcus, it is caused by E. coli, various number of bacteria. Large number of bacteria causes mastitis. Okay. So this disease in bovines is caused by the following streptococcal species: S. agalactiae, S. dysgalactiae, S. uberis. These are the three main species. You have to learn these three only. Principal pathogens. Now, Streptococcal agalactiae causes acute mastitis. Streptococcal dysgalactiae causes acute mastitis, and Streptococcus uberis causes clinical mastitis. Okay. Now, diagnosis: clinical sign: inflammation of mammary gland or clot in the blood milks. Blood milk clots in the blood milk. Blood milk clots in the milk. Sample should be cultured on blood agar, Edwards medium, McConkey agar, incubated aerobically at 37 degrees Celsius for 24 to 48 hours. Now, tests done for diagnosis: sugar fermentation test, SFT, PCR, and positive camp test. So what is this? This is important. Positive camp test. So Streptococcus agalactiae releases a factor which completely lyses the red cells. Okay. So Streptococcus agalactiae, it releases a factor which breaks down the RBCs, and these RBCs already damaged by beta hemolysin or Staphylococcus aureus. So this completes the hemolysis produced by Streptococcus agalactiae, produces a characteristic clear arrowhead pattern of complete hemolysis. So this is the basic meaning of camp test. So